this video we're going to be looking at proteins. This is another one of our macromolecule examples. You can see from the picture that proteins come in a wide variety of shapes, wide variety of sizes, and they bond differently. There's a lot of creativity when it comes to proteins. They're made up of these four main elements. Occasionally you'll see sulfur in here as well, but the main elements are carbon and hydrogen and oxygen, which we had with our lipids and our carbohydrates, but now we're also adding nitrogen. So nitrogen's kind of the new element there. That is what makes protein a little different than the carbs and the lipids. Proteins have multiple jobs. One of their jobs is that they help speed up chemical reactions. So they make reactions that normally would take quite a while in your body to maybe break something down or to build something up would take quite a while, but with a protein they can make that move quicker. They also provide structural support. You can see that there are proteins here embedded as part of your cell membrane, so those proteins have a part in forming your cells. They also help store material and transport material. As you can see here, those proteins that I circled up above, if we look at a different picture, you can see that the proteins are traveling through to get either inside or outside of the cell, so they do help transport. Cell communication, they help with movement, and they also help defend against foreign objects like antibodies that you have in your body that help defend and protect your body would have quite a few proteins involved there. Enzymes will be a whole separate video of its own because enzymes are the biggest example of proteins, and these are the ones that speed up reactions. And that's a really huge one. So enzymes are proteins that speed up reactions, and when we talk about an enzyme, these are examples of catalysts. A catalyst is any chemical that speeds up a reaction. So all enzymes are catalysts, but there are several catalyst examples that aren't proteins, that aren't enzymes. Here's an example of a catalyst, this green object here. And if we were to take two chemicals from your body that need to get bonded together, it may take them a while to get bonded together on their own, but with the help of a catalyst, each one kind of finds its spot that it fits into, and then the catalyst creates that bond, helps them to bond together. So it just speeds the reaction up. Every protein has its own shape. They're three-dimensional. They're not flat. They are three-dimensional. They're very unique. We also have mentioned for every one of our macromolecules what the monomer is. Proteins are polymers. They're made up of individual items that are bonded many times. And the monomer for a protein is an amino acid. There are 20 different amino acids, and what makes all the amino acids similar is that they have this amino group. So they have this, that's where the nitrogen comes in. The nitrogen's bonded to some hydrogens. And they also have these other pieces, but every single one of them has this amino group, and that's what gives them the name amino acids. Now, when two amino acids are going to connect together, they form what we call a peptide bond. And you can see dehydration synthesis at work here as well. An oxygen, and we have two hydrogens, are being removed, and that creates this bond here that they've labeled a peptide bond. And you also have the water, so we have the dehydration synthesis. We know our prefixes. Poly means many, so literally a polypeptide just means many peptide, and it's referring to the bonds. So if you were to do this again and bond more and more items together, you could have like 100 amino acids bonded together, so you would have a lot of peptide bonds, so we call it a polypeptide. Polypeptides can vary in length, just like you have monosaccharides, which are 1, and disaccharides, which are 2, polysaccharides, which have many carbohydrates. That's the biggest, many sugars. You can have a polypeptide, you could have 100 amino acids, you could have ones with thousands of amino acids. The function of the protein depends on what it recognizes. We said that each one had its own unique shape, and so you can see they're three-dimensional, kind of like puzzle pieces, and whatever chemical it reacts with has to connect and bond to a certain location, and we'll get into that with the enzyme video. But the shape of the protein is important because it won't bond correctly if the shapes don't match up. So that's how it recognizes what it should bond with, is by the shape, and then it can do, the action doesn't actually happen until the two are connected. An example of a protein that you would have that is incredibly important is hemoglobin, and this is the protein in your red blood cells. So if we were to take a look at a little sample of a red blood cell here and magnify it, you have this twisty, turny, three-dimensional object here. That's the hemoglobin. And these 4 blue balls represent oxygen. So your hemoglobin is how the oxygen gets carried on the red blood cells. Every hemoglobin molecule should be able to carry 4 atoms of oxygen at a time. Now, if the shape of the hemoglobin were to change, and there are some genetic mutations where the hemoglobin doesn't have these 4 open spots, then it can't carry the oxygen as well. And an example of that would be this type of red blood cell, this one that has what we would call a sickle shape. It's called a sickle cell disease, sometimes referred to as a crescent shape. But because the shape of the red blood cell has changed, it's changed because of the hemoglobin. And it can only carry 2 oxygens at a time. So someone with sickle cell might have less energy because they can't hold or carry as much oxygen as someone with a normal shaped red blood cell. Lastly, for today, we're going to look at denaturation. Denaturation is when a protein unravels, and when it unravels, it loses its shape. So if it loses its shape and it can no longer, this is the spot where your other molecule is supposed to come in here and it was supposed to bond. Well, if that shape doesn't fit anymore, then you can't bond with your other chemical. And so that means that the reaction can't happen. So that active site is important. The shape of the protein is important. Denaturation is bad. It causes the protein to not work or not work as well. Some examples of things that would affect the protein: if the pH, if you have something basic or acidic, that could affect the shape of the protein. Salt concentrations and temperatures, probably the biggest one. For instance, if you've ever made an egg in the morning, all of this that used to be very clear material is protein. And when you heat that up on the stove, it turns this white color, and that's because the temperature is taking the protein of the egg and it is changing the shape of the protein, hence then changing the color as well. It's a characteristic that changes. So that's not a good thing. The protein doesn't work the same way it did before. And denaturation is something we will also be doing some labs on so that you can see some examples of how that works.